Hello to anybody who's going to view this in the future. This is a conversation between Dr. Martin Jonikas, who is an assistant professor at Princeton. He has been a superstar or a rising star in the field of algal biology. And the interviewer, that's me. I am a postdoc with Rebecca Roston at University of Nebraska, Lincoln. So the structure of this interview or a conversation is more focused around Dr. Martin Jonikas's work style, his mentorship style, and other stuff. It's not pertaining with his work, which is, by the by, a really fascinating a bunch of work that he has done. So with that, I thought that let's start the conversation with Dr. Martin Jonikas from Princeton. So I wanted to start with a very, very pertinent question for now, for these times. How are you coping up with the COVID-19 lockdown situation?
Thanks, Ritesh. We're fortunate to be healthy, and we're on lockdown, and we're doing the best we can during this time.
So how are you keeping up with the lab businesses and your teaching responsibilities?
So I'm fortunate that I don't teach in the spring. I teach in the fall.
Okay, that's a blessing.
But so we'll see what happens next semester. But for now, we're trying to keep the lab productive as we can while being locked down. Our strains are being propagated by two very dedicated people in the lab, and meanwhile everybody else is trying to do what they can from home.
Yeah.
For many people, they can be productive because they have data to analyze, and others are planning future experiments.
Yeah, that's a reasonably good thing to do. And we were talking the other day with one of my friends that the only bunch of people that I know whose work cannot be stalled are the algal folks, because the yeast folks can put it in the freezer. The mammalian people, cell line culture people can put it in the -194, but clammy or any other algae, that's a tough gig to do. Yeah, so my first actual question is that you are an MIT trained aerospace engineer. How did you move from engineering from MIT into a cell biologist, a geneticist in general? How did that transition happen?
Well, I've always been fascinated with how things work, and also with things. Machines that do things that should not be possible. That's how I was drawn into aerospace engineering. Flying things kind of defy our intuition for how things should work.
Yeah.
And so I've always and still am fascinated. I've always been and still am fascinated with flying things, and with I started out as a kid flying remote controlled airplanes, and that's what drew me to aerospace engineering. And I really enjoyed studying engineering, and I just by chance during my second to last year of college, I was forced to take a class in molecular biology, and I just had a really fascinating professor, Joanne Stubby, who just kind of blew my mind about how amazing biology is, and showed us how biology is made up of all these little machines that do amazing things. And I was really inspired, and decided that was a direction I wanted to go.
I mean, like, she should have been a really great, professor to at that penultimate stage to push you towards life science. Yeah, that's really a really interesting thing. Then there was another transition, which is not that drastic, but it's still drastic. You were an yeast geneticist. From that luxury of being an yeast geneticist, you moved into Chlamy, which I'm sure you would appreciate is not it's not the most hard, but it's not the easiest either. How did that come about?
Yes, so also somewhat by chance. Some of my classmates and I, as we were getting close to the end of our PhDs, we started a journal club where we were pitching different ideas for what we could do as our next steps in our careers.
Yeah.
And so one of my classmates pitched this idea of kind of this ongoing excitement about biofuels and photosynthetic organisms about 10 years ago. And I just, it blew my mind again that I had never realized how important photosynthetic organisms were to the planet. And
Yeah.
I started reading more about them and got very excited about them. And I started thinking about how I could contribute. And I knew nothing about photosynthesis, but I had just trained as a yeast geneticist. And so I started to think about how I could apply the yeast background to photosynthetic organisms. And that's how I became very excited about Chlamydomonas.
Yeah, and then that's quite reasonable. That's pretty much how I also got into that around 10 years ago, or 10 to 20 years ago, when the buzz of algal biofuels was on a fever pitch
Oh,
status.
really?
Yeah, I mean, like when I was in college. So then the next step for you, to me at least, it seems like it was quite a risky move that you took. From grad school. You went to a PI position, and you took on an organism that you didn't work with, and an organism which is tricky, and you took on a project which is quite remarkable. You started generating the mutant library for Chlamy, which had not been thought of, at least for the 50 years or so. How did you muster the courage for that?
Well, I would say I was very naive. That's
Naivety is sometimes good.
yeah. So we, and I was really fortunate that a lot of really talented people joined my lab and really pushed the project forward. We had this very naive idea that part of the power of yeast genetics was these mutant resources that allowed you to study the function of any gene.
Yeah.
And there really wasn't such a resource available for a single-celled photosynthetic organism at the time. And so we thought there was a big opportunity there. And I think there was, but we just didn't realize how hard it was going to be. And we were just really stubborn and naive, and that's how we made it happen.
And I think as a field, algal biologists would be happy that you did that. Then the next project that you took on was this interactome, which again is not a cakewalk. It's not the easiest. It is a complicated project. So my question is, how do you convince postdocs? I mean, like, these are projects which are labor intensive. There is no absolute certainty that you are going to get something great out of it. How do you convince your people to join in on these risky ones?
Well, it certainly takes a certain kind of person to take on a project like that. People have to be interested in and believe in the value of these types of resources. But I think in many ways, these types of projects are actually pretty safe and pretty likely to be impactful because we have seen how valuable they have been in other systems like yeast and Arabidopsis. So they're primarily technical challenges of getting them to work. But once you solve the technical challenges, you can turn the crank and produce something useful.
So first it was the library generation, then it was interactome. What's your big thing that's happening in the lab right now?
Well, the lab is really working on two areas. One of them is this large-scale biology, and the other is the pyrenoid.
Mm-hmm.
In terms of large-scale biology, we're taking the things that we've started and we're actually taking them to the next level. So we are starting up um, both a project on high throughput genetics and a project on protein localizations and interactions. In terms of the high throughput genetics, we are trying to determine a phenotype for every gene in Chlamydomonas. And we're trying to use these phenotypes to place genes into pathways. The dream is to essentially figure out how all the genes in the genome are organized into pathways and thus predict functions for uncharacterized genes.
So
In
that,
our,
yeah.
sorry, go ahead.
So that's kind of the functional genomics at the true forefront.
Well, we hope that it will be useful. And it certainly has been on a small scale already. Yeah, and these other projects, we're trying to systematically determine the localizations of nearly all proteins that are associated with the chloroplast.
Oh.
And then determining their protein-protein interactions. And obviously, this kind of information has been incredibly useful in other systems like yeast and humans. And so we're hopeful that this information will help really advance our understanding of protein functions in photosynthetic organisms.
Well, I certainly would agree, because when working with algae and when you get a transcriptome data, a lot of genes have unknown as the description. So it's a little bit annoying. It would be really good to know something about those fellas. So how did you then get into carbon concentrating mechanism? Because that's a field which has been worked for at least the past 30-ish years. How did that happen?
Yeah, so we again, also this happened by coincidence. I knew that I was interested in photosynthesis. So when I first started the lab, I knew that I wanted to develop these tools, these resources, but I didn't really know what problem to apply them to. And I knew so little about photosynthesis that I didn't know where the big opportunities were. And so I was really fortunate to be invited to a conference that was organized by the National Science Foundation. It was a so-called Ideas Lab, where they brought together a bunch of PIs from different backgrounds and different levels of kind of different stages of their careers. And we brainstormed different ideas for improving photosynthesis. And this is where I got introduced to a lot of exciting directions. At the time, actually, it wasn't clear if photosynthesis could be improved. It was seen as this thing that maybe had already been optimized by nature. And since then, over the past 10 years, we've seen a lot of examples where people seemingly have actually been able to really enhance photosynthesis by a number of different approaches.
Yeah. I mean,
So,
like, a lot of focus was on Rubisco, the giant, and many other things were left out. Yeah.
Yeah, so actually, interestingly, so out of this conference came out a number of projects, and I think it was something like 4 projects were funded jointly by, it was actually a joint effort between US labs and British labs. And they were funded jointly by the US National Science Foundation and the British equivalent, the BBSRC. And so we formed various teams of international collaborators. And on my team were actually 3 British labs, one of whom was Howard Griffiths, who was a researcher working on the algal CO2 concentrating mechanism. And he actually came up to me during the conference and he said, it would be great if we could apply your tools to this problem. And he was right, and there was a huge opportunity there, and it's really thanks to him that we've entered that field.
So what's the a 5 years goal or a 10 years or a throughout goal for you in CCM?
Right. So there's the CCM is important for two reasons. So one of them is essentially a large fraction of global carbon fixation, about a third of global carbon fixation, is mediated by algae in the oceans, and by this CO2 concentrating mechanism that helps them essentially suck CO2 out of their environment.
Yeah.
And so merely studying these CO2 concentrating mechanisms and understanding how they work will advance our basic understanding of how the planet works.
Yeah.
So that's one opportunity. And the other opportunity is we have this crazy dream of trying to enhance photosynthesis in crops by transferring innovations from the algal CO2 concentrating mechanism into crop plants like wheat and rice. And there's a substantial amount of solid science that suggests that if successful, this kind of a transfer could increase yields by up to maybe even 60%, and the crops would require less water and less nitrogen fertilizer.
Yeah, and also climate change affects in any one of these ranges. Yeah.
Yes, it could also benefit climate change.
Yeah.
So we're really dreaming of understanding the algal CO2 concentrating mechanism to a level where we could actually engineer it into plants. And so our goals, let's say for the next 10 years, would be to actually, and I should say this is part of a larger international collaboration. Our goals are to engineer the algal CO2 concentrating mechanism into land plants and see improved crop yields.
Hopefully that bears fruit sooner than later. So moving along to your style of being a PI, boss, mentor, all those things. For yourself, how do you manage your class, like, your workload with concerning class, your mentorship, your writing, and all the other things? Do you have a style that you follow?
So it sounds like maybe there are two questions there. One is about time management, and the other is about mentorship style. Is that right?
Yeah, there is a separate question on mentorship style later on. So if you want to
This one is
answer,
about
the
time
time
management.
management should be fine. Yeah.
Yeah. Well, time management is something we all struggle with, and we all have a lot of opportunity to improve. We can always be improving, and I'm still learning how to make the most of my time. But it's true that we all have one limited resource, which is time, and we only get so much, and we have to make the most out of what we have. So certainly investing in I try to invest in learning how to make the most out of the time that I have. So some of the things that I find useful is, in general, investing time into actually planning to make sure that my time is spent on the things that matter. So that means first figure out what are the things that matter, and then have a framework for making sure that my time goes there and not to all the other distractions. And so I use things like Excel spreadsheets, to-do lists, and a calendar. Every week I spend about half an hour to an hour reviewing my priorities and taking the highest priority items and putting them into my calendar. And then for the rest of the week, my calendar runs my life.
That's
So I don't
a
have,
good strategy. I mean, like I've heard many people give suggestions, but yeah, never thought about spending time on deciding how to spend time.
well,
Uh,
yes, I mean, when you think about it, if you don't, uh, if you don't rationally decide where your time is going to go, then there's all sorts of things that want our attention. I mean, I'm not saying that it's perfect. I still get distracted.
Yeah, I mean, like when you are, I've heard that when you are an assistant professor, I mean, like although it might be a little less of a transition for you, but when you're an assistant professor, a whole lot of things is being shot at you, and that might be a good strategy to like manage your time to manage your time. Um, so that
Sure.
then comes to the next question about your mentorship style. When I actually went through your profile, probably like 3 4 years ago, um, or actually even recently, the most surprising thing to me was that all six of your postdocs who have left the lab have all landed really good positions, which is kind of remarkable, or at least to me, it seems remarkable. Um, so let's talk about that. What's your mentorship mantra?
Well, um, uh, let's say I think that probably the biggest reason why these postdocs were successful is that they were all outstanding, um, future faculty essentially. So I think the key to having people move on to successful next steps is to have them to hire people who are already on that trajectory, and they just need a little bit uh, of nurturing environment uh, to 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 reach that stage. And so I've been really fortunate to have a lot of really talented people uh, come through my lab, and I do the best that I can to help them move on. And certainly, it is a high priority for me. It's a very high importance, and has been from the very beginning, to help everyone in my lab uh, move on to um, their next step in a position that they would like. That has been a very high priority for me, and uh, I said that as a high priority because I know that if uh, new people who are considering joining the lab, if they look at our track record and they see that they will evaluate us
Mm,
primarily based on how other people have fared through the
yeah,
lab.
that's a really good metrics. And so is there something that you do, like a practice that you follow, like anything that you think that there's like, oh, I follow this one, I think this is really helpful in being a or nudging them to move forward?
So this is, I'm not sure if I understand the question. This is a practice for helping people. Um,
I mean, like the really talented folks come into the lab. I'm sure there would be something that they need to, change tone or something. How do you identify that for them, or what sort of practical helps do you give?
um, I think by the time that people come to the lab, they are largely
Hmm.
formed. I mean, people who are, especially who are postdocs, they, I don't know, they're like, 27, 20 to 30, or
Or
so on,
31,
or
32?
31. And, by that time, you've really formed who you are. And
True.
so I can't really pretend that I fundamentally change anything about people,
Mm,
but I do try to provide them with a nurturing environment that um, provides them with resources and opportunities, um, hopefully, the environment is also includes other smart people with
Yeah.
whom to work and collaborate. And I try. Okay, so I'll say a few things. So one is I do care about the environment. I try to nurture the environment, and I think so. Some of the things that I try to do is try to hire nice people, and so not only are people smart, but they also they're nice and they interact well with each other, and that I think is very important, because if you're in an environment where other people are nice, then you're nice, and it's kind of
Sure.
a positive cycle. I also try, science is hard, and often, there are daily things in our scientific lives that cause stress, like your experiment might not work out, or you might accidentally mess up somebody else's experiment. I know I have
Yeah.
when I was a PhD student, and so the atmosphere and the kind of the culture of the lab really matters a lot
Yeah.
when those types of things happen. And so I try really hard to nurture kind of uh, people getting to know each other as people and not just other people in the lab.
Yeah.
And so we do things like we have the lab has lunch together every week,
Oh.
once a week, and we do various lab outings, like we go to dinner to celebrate special occasions, and we go on various trips, like hikes, and we went to an amusement park once, various things like that. So these are kind of social interactions where we're really not necessarily talking about science, but we're just getting to know each other as people. And so that's kind of like a preemptive investment in people's
Yeah.
friendships, so
Yeah.
that when those rough moments come up, they're not attacking a
Alone.
total stranger for messing up their experiment. They can understand it's another person. So I think that's an important aspect.
Yeah.
Yeah.
So, so you did touch upon this, but I'm going to try to frame this as a question. So, what are the intrinsic qualities in these postdocs that you think has helped them really being successful? May that be their smartness or other thing. Also, what do you look for? As a scientist, what do you look for when you're hiring a grad student or a postdoc?
Um, well, of course, it's very hard. It's extremely, I find hiring, I mean, obviously hiring, I think, is probably the single most important decision that any kind of team
Mm,
leader makes. Um, because really, the success of the team is really based on the individuals
yeah.
who participate in the team. Um, I try to look for a number of things um, in the CV. I try to look for um, examples of past productivity, and this usually means at least one uh, first author or co-first author publication. Um, I also um, look at the cover letter. I look at whether they uh, exhibit an interest specifically in our lab,
Mm,
um, um, or whether they're just blanket applying.
mm,
Um, I try to look for some uh, sign of passion. I
yeah.
think passion is very important. Um, of course, it's very hard to do that via paper. So really, once I kind of have screened through these first few things on paper, um, I try to reach out to the references,
Mm,
because the references know uh, the person. I mean, much better than even I can get to know them over several days
yeah.
of Skype or in-person interviews. And I really try to get a sense about the person via from the referees. And I find that to be particularly helpful to gauge how good of a fit they would be for our lab. And then we also, of course, invite people in person to visit the lab, because, and those are people who are very likely to actually get an offer in the end, because I think it's very important for both for the person to get a sense of what the lab is like,
Yeah.
because it's an important decision that they're making, and also for the lab to be able to provide input on how well they feel the person can fit in the lab. And then the other thing actually that I do look at is the I have the candidate give a presentation, and I think the quality of the talk is pretty informative as to how the person thinks about their science, and what the person accomplished. And also, I actually think to myself, like, would this talk, like based on the person's PhD work, make them competitive for a faculty position?
Mm.
Right?
Okay.
So essentially, like, if they're already, and what you're ideally looking for is someone who's already competitive for a faculty position, and you're just providing them a home to check that box that of doing a postdoc.
That's a really good logic, and which has a very fair amount of certainty locked in there. That's really a good one. Um, so, sticking with the mentorship, a question which generally comes up in all of these kind of interactions: are there mentors apart from your undergrad professor? Are there people those who you consider are strong mentors or have had a really good influence on you in being a scientist?
Yeah, so I've been incredibly fortunate with mentors, and I guess really I wouldn't be anywhere without actually the very many people who have mentored me, and yeah, I mean, obviously, I think my PhD time was extremely kind of influential, because I came to do my PhD having no biology background, and so there I was really fortunate actually that by chance I ended up in a really wonderful and amazing environment at UCSF. I was in the lab of Jonathan Weissman, who is an
East, big
incredible
shot, yeah.
scientist. And then I was actually co-mentored and jointly, uh, also jointly advised, and a joint student with Peter Walter, who's another amazing scientist. And then in the lab of Jonathan Weissman, I was mentored kind of on a day-to-day basis, because I didn't know how to do anything in lab, by by Maya Schuldener, who was a postdoc at the time and is now a very successful scientist, or a PI at the Weizmann Institute in Israel. And so I was just super fortunate to land in it, and it was completely by chance that I ended up there. I was just really fortunate to be mentored by these three amazing people, and I think from each of them, I learned different aspects about how to do science, and I'm really grateful for that.
I'm pretty sure that you have brought laurels to all three of them, by being an amazing scientist. So another interesting catch in your career, which is completely, again, another level of unconventional situation, is that you did not do a normal postdoc or a conventional postdoc. You went on to become an independent PI, and again, took on risky projects. What are your, looking back and retrospecting, the good and the great and the not so good aspects of taking that route?
Well, I was, I think I was incredibly fortunate to have had that opportunity, and specifically to have had it in an extremely nurturing environment. So I started immediately out of my PhD. I had the opportunity to start a lab at the Carnegie Institution for Science on Stanford campus, and it's, in hindsight, it's completely crazy that they took a chance on me, because I was fresh out of my PhD. I barely knew biology, because I'd been doing biology for like 5 years, 6 years, and then second of all, it was a plant biology department, and I was proposing to do plant biology, and I knew nothing about plants. So I have no idea what they were thinking, but they were just they took an amazing chance on me, and um, uh, on top of it, they really nurtured me through it. And I think I owe a lot to the faculty there, and I think the director, Wolf Frummer, was just incredibly nurturing to me, and nurtured me as a as a young scientist, and taught me how to apply for grants. And actually, when I first started there, it wasn't clear if I was going to be successful. Like, I guess for anybody, it wasn't clear if I was going to be successful um, in applying for grants, and there wasn't even that expectation that I would apply for grants. But Wolf gave me the opportunities to apply for grants, and encouraged me, and kind of gave me advice on how to do that. And then we were successful, and so uh, it was just an amazing kind of nurturing environment. Also, we had a close colleague. One of the reasons why I went to Carnegie, uh, even though I knew nothing about photosynthesis um, or algae, is that um, uh, a world-renowned expert in algae, Arthur Grossman, was right there, and in fact was our next door neighbor. And so we we had a lot of wonderful interactions, and a really great collaboration on developing this mutant resource. And I also learned a lot from interacting with him and his lab. And it was just an amazing nurturing environment. Yeah.
Sort of, I mean, like, I don't know if I can call that serendipity, but again, even serendipity only works when you are smart enough to make it work. And you seem to have made it work very well. So do you recommend that route to grad students at all, or do you think that it's too risky?
Well, I think it's I in hindsight, like, we were extremely naive in how, like, I was extremely naive, and I spent the first I was not mature enough, I think, in many ways, to run a lab. Like, we spent probably the first 2 years focused on, like, nitty gritty details, trying to make things perfect that didn't matter. Um, so we made a lot of mistakes that I think we wouldn't have made if I had done a postdoc. Um, at the same time, because the environment was so nurturing, we got past that stage, and we're able to become productive. And I think because there weren't the pressures of tenure as there would be in most institutions, it was okay that we took on these incredibly risky projects, and it was okay that we weren't successful initially. And this I think this nurturing environment gave us the opportunities to succeed in these really ambitious projects. So I would say it's primarily a question of the environment that you would go into. I would say definitely a postdoc with a lab that has a track record for launching people. If someone is interested in going into, say, academia or industry, actually, going into a lab, doing a postdoc with a lab that has a track record of launching people into such positions is definitely a pretty good bet. Trying to start a lab straight out of your PhD is much more risky, but at the same time, it worked out for me, and it also worked out for many other people who have done these types of transitions. Actually, the vast majority of people who have done these types of transitions, and it can be also it can be a really wonderful experience, and so.
Yeah, I mean, if I'm not wrong, Broad Institute has invested into things like this. Stanford has started these programs. Carnegie has always done this. That sort of renders my next question moot, which is what would be the suggestions that you would give for graduate students, those who want to, skip that or actually keep the postdoc period as short as possible? But if you want to comment on that.
Yeah, I mean, I think the most useful thing to me was to write a proposal for what we were going to do. So as part of applying for these faculty positions, I had to write proposals, submit proposals, and in the process of writing the proposal, you plan out, you produce a plan, and you hopefully you understand why the work is important, what the challenges are going to be, and how you're going to get there. And the plan that I still look back on the plan that I wrote in applying for these positions, and there were 3 aims, and a large fraction of the lab has been working on 2 of these aims for the past 10 years. The 3rd aim we never got to work, but that proposal provided guidance for the lab, and was incredibly useful thing to do, to write. Of course, new things came along that we would have never expected, but having a plan was extremely helpful.
At least it'll make the wandering a little more engaging. A plan. We are almost there. Do you have any words of wisdom or things that you learned for graduate students, postdocs? Although you didn't go through a postdoctoral training, but still, and for assistant professors, because you have an experience of running a lab even before being an assistant professor.
Well, I would say that one aspect that I never expected, I don't know if this is a word of wisdom, but it's kind of one aspect that I never expected going through science and leading a lab is how large a fraction of what we do is actually interacting with people. It's actually, I went into engineering and then into science thinking that I could be in my own little bubble and just do technical stuff. But actually, as you move through science and start leading a lab, you realize that actually a very large fraction of what you do is communicating with people, hiring people, helping people work through challenging situations. You yourself have to, how you communicate things matters a lot. And so these are all things that don't come naturally to me. And I've just, it's been a twist. I think a very enjoyable twist, actually. It's one of the things that I enjoy the most about our line of work is actually getting to work with really amazing scientists, both in the lab and also outside our collaborators, and just rubbing shoulders with amazing people in science. So I've just found it really fun and interesting to start learning a little bit better how to interact with other people.
So were you an introvert basically?
Yeah, I don't. I guess I was an introvert. Yeah, it's been my through college until college. I guess I was really kept to myself. Yeah, so this has been a journey.
uh, Thank you, Dr. Jonikas. It was really engaging to hear your perspective and to have interviewed the Elon Musk of algal science. And I hope that you and your lab are able to break barriers and achieve great things. And hope to see you one day in person, and probably once of all this COVID shenanigans is settled down. Thank you for doing this.
Thank you so much, Hitesh. I really appreciate your time and this opportunity, and I do hope that we can all get out of this really unfortunate situation quickly.
Hopefully, soonly. Thank you. Thanks a lot.
Take care.